the contents into today's class, and I put up some material, some URL links to YouTube videos that will help your understanding. I hope you can go through all the videos and understand, especially on action potentials and how sensory, how the how does our nervous system relates to our other part of our bodies like muscle and so on. So there's one example on muscle construction. One video on how our nervous system interact with muscle to produce muscle contraction. Okay, so at least you can get an idea on how this neuron affect the whole body and in a way give instruction to our body to move or react. Okay. So today we are going to look into neurocoding, coding techniques and information theory. So we'll start first with neurocoding. So of course in neuroscience we would like to understand the function of neural circuits in the brain, how they are related to behavior. And there are a lot of efforts that have been done in previous years to develop techniques that enable recording of the brain and electrical activities at different spatial scales. All these EEG, the invasive and non-invasive methods, that we learned earlier. And this concept of measures ranging from localized sparking activity of individual neurons, which local field potential, and all the way up to the EEG. What is neuron coding? Neuron coding is a way to understand how the firing of neurons in the brain leads to execute its brain function. So how this neuron, the action potential actually cause our body other response that relates to other part of the bodies via brain instruction, from sensory processing to execution of motor commands. So from stimulus means whatever we see or whatever we feel, we touch. Feel means based on mind, but feel means based on sensory, like what you touch, like reflex when you touch hot spot, then you think you sense hot, then your brain automatically asks you. So from that, the moment of you sense the hotness, till you pull your hand, there's this firing of neuron is happening throughout the cycle. So from accepting the stimulus to the response. So what we look at, we usually measure neural response rather than stimulus. So when later you'll see what are the what in detail about that. To explore what the code that makes us aware of our own existence and everything we do. And the main component of neural code is the sequence of action potentials emitted by individual neurons. Okay. From this data, we need to extract firing, perform the firing. Okay, we need to do data extraction so that we can extract the firing of each recorded neuron. And this procedure is called spike sorting, which separates the spikes from each neuron according to the shapes. And this, if you are working, since we are working with a lot of neurons, so today, thousands of neurons, so we need to have this amount of that. This is a huge number, so we need to have unsupervised and fast algorithm. So once the firing of each neuron has been identified, then we can actually interpret the activity of dozens or even hundreds of neurons and code. So, right. So, what we need to know when we do in neural coding, we need to understand what is spike. Okay, spike is basically your action potential Okay, so uh, okay, okay. So neurocortex projects its own output via sequence of spike called spike train to other thousands of other neurons. So this spike train is actually your action potential. So it says a spike is action potential. A spike train will be a sequence of action potential. So a spike train is a sequence of recorded times at which a neuron fires an action potential. When the voltage drop across a neuronal soma or axon membrane is recorded. The pulse of roughly 100 millivolts, and these are action potentials or spikes. Okay, so in order to observe neuronal activity in the active brain, uh, many recording techniques are being employed, ranging from intracellular to extracellular. Okay, so this is how recording being made. Okay, so please bear in mind that this is not being made in human. Most of these EEG experiments are being made on animals, not on human. So some recording methods they use electrode that goes deep into the brain and right to the neuron to the what, to the brain slice or isolated neuron. So they go deeper into inside the brain, the animal brain and so on, they put the electrode there and the reading will be different. You can see the intracellular recording and the extracellular recording. Um, okay. So intracellular recording and extracellular recording are different. You can see the difference. So where where the position of the electrode give different type of uh, spike, okay, or spike pattern, okay. So remember, action potential is actually spike, and sequence of action potentials is called spikes. Okay, so the firing rate is related to the number of spikes generated by a neuron per unit time. And the phenomenon in which fire rate vary with stimulus is called rate coding. So, if you have a strong stimulus, okay, your neuron may produce a higher firing rate, stronger firing rate, or faster. Uh, yeah, you can, or if it's weaker, then it will be slower. So it depends on how strong is the stimulus. So, for example, if you compare touching a gentle surface which have no harm, and touching a hot pot which is harmful, which one would have a stronger fire rate? You think would have a faster firing rate? So which one do you think? Oops. Hi. Hello?
Yeah, doctor.
You guys with me?
Yes, doctor.
Okay. So which one would have faster firing rate?
The hot pot.
Okay, the hot pot. Hold on, I'm trying to find your one. Okay. While analysis of neuroactivity in terms of verifying rates has been defined earlier, okay, it's very useful in many a more subtle alteration of the pattern of spike times also occur in MA content information. So pattern does give a lot of information. So for example, the length of gap between spikes known as inter-spike intervals. So this often vary substantially across along the spike rate. So each spike rate for every response they will have different spike frames. Okay, and this can also be observed through the gap between the spikes, interspike, and some specific percept or motor action would have some will be represented by the elevated firing of a single nerve cell. So this is called estimation of neuronal firing rate. Okay, some of the neuron may fire, when the spike, while others in the spike train nearly similar firing rates, but some suddenly some of the neuron will have higher firing rate. Okay, so when the signal goes through them, so that's called estimation of neuronal firing rate. Okay, so this is the spike sorting algorithm. Okay, so this is the original um, this is the original extracellular recording, and this is then the action potential is being instructed. So you can see the spiking here, and then you perform noise reduction, and this is where you detect the spike, and then you start to do spike extraction and alignment. Okay, so with the timing and so on, and feature extraction and spike sorting, identify spike. And this is your raster plot for your spike frame. This is called pre-process. This in computer science, this is pre-processing, and this output will be used further in for data mining or machine learning. Later we have that section. Okay, spike frame statistics needed when in noisy environment, regardless of in vitro or in vivo. Okay, in vitro is when you put when the electrode is inside the brain. In vivo is when it's outside. It's non-invasive. So where we let it record the whole brain function, not just a neuron. Measurement of output input and state, and what kind of code that we need for spike frame produced by spike frame statistics. Rate code inputs which look at bunch of spikes, and spike time which is actually a temporal coding which look into individual spike. So how to distinguish between rate and temporal coding in practice? When precise spike timing or high frequency pyramid catch rates are found to carry information, the neural code is often identified as temporal code. So the spike carry information. Okay. The temporal structure of a spike rate or pyramid is determined both by the dynamic of the stimulus and by the nature of the neural encoding process. So if there is an interplay between stimulus and encoding dynamics, then it makes the identification of a temporal code difficult. So that's for temporal. So this is example of temporal coding. So basically we do spike count. So if depending on different stimulus, the response will be also different. So if you see this shape, then this response is like this. So the spike count, and what they did is they convert, okay, where they see the stimulus, the brain produce this EEG the signals. So this is your EEG plot. Then it's being processed into the response. This is the response. Is being processed into temporal based on temporal coding into spike count. Okay, so what happened is the signal, the spike is being identified based on the time. So and then you get a binary representation where it comes. So this, if you could see that if they pick, and this representation is being means into a specific length of representation. So you can see all these responses have the same length. So each bit position is referring to the same timestamp, or not timestamp, or time gap. Okay, so for the first, you can see for when you look at this stimuli, this is the responses where there are 4 spikes and it appear at different time. Yeah, as compared to the cloud and the smiles. So they have the cloud have 3 spike, and the spike appear in different times as well. Okay, so these are pattern, and this is the spike count. So this is temporal coding. So the overall process, there are 2 ways of estimating firing rate. Okay, one is non-parametric methods, and the other is parametric methods. So we have a monkey here, so we put the electrode on the monkey brain and let it see some pictures. So we put apple and banana. So and then when the apple appear on the screen, we record the signals, and we can use parametric methods to produce this firing rate graph. Okay, so but it has to be repeated trials. You cannot take this on one time, at one time only. So it has to be, the apple has to appear a few times, so that with it has to appear in rotation with other pictures, so that we can get a precise pattern for this apple the sensory for this apple. Okay, so this is still and then so when it sees apple, it will give some pattern or some spike patterns, and we can calculate the firing rate using PSAH, Paris stimulus time histogram of the low density function. So information is in the difference of firing rates over time. So this graph will be the representation of the spike for the stimulus onset, which means that when the stimulus is active. Okay. So from here, we can actually later we can use this spike, this diagram, or this pattern to identify in a larger set. We can use it for data mining actually. Okay. Right. So this is for banana, and this is how the spike diagram looks like with different firing rates. So you can see the different firing rates for each picture. That's non-parametric methods. Parametric methods is based on probability. Okay, so rather than producing firing rate over time, they come up with probability distribution. Okay, so this probability distribution is based on parameter estimation methods. So the P that they are later you see in the next slide. So the methods are maximum likelihood, maximum posterior, and expectation maximization. So I'm not going to go through all of this method in detail. I'll just go through the notation on probability. and some important points for what I call interpretation. I'm not nothing to do with parametric method, but just some notation for that is important for the interpretation. So models based on distributions, definition, and so it's just like definition and symbols. So what we measure is the probability of an event spike, and the probability density function comes from whatever method you use. Okay, so probability corresponding to every single spike that can be invoked by the stimulus is given by some equation. Okay, so what you need to know is about the discrete random processes that happens in an interpreting spike patterns. So it's whether it's a point process, renewal process, or Poisson process. Point process is when the probability of an event could depend on the entire history of proceeding events. Okay, and renewal process is the dependence extend only to the immediate preceding event. So point process is when the probability of one event, the current event, um, that depend on the future event. A renewal is when the current event depends on the immediate on the previous event. Poisson process is when there is no dependence of any all from the preceding event. So no means independent. So the firing rate of a single spike is RT in a small interval around time I is not generally sufficient information to predict probability of spike sequence. Okay, so if the probability of generating a spike is independent of the presence or timing of other spike, then the firing rate is all what we need to compute the probabilities for all possible spike sequence. Okay, so this is easier lah if we use parametric. Was it non-parametric? And then homogeneous distribution is when the firing rate is considered constant over time, and inhomogeneous distribution when time is the firing rate is time dependent. So Poisson when each event is independent of others. And again, the homogeneous is when the probability of firing is constant during the period T. So each sequence probability have similar. Each sequence will have similar probability lah, because it's constant. Interspike intervals distribution or ISI. Okay, so the probability of the density of tangible between edges of the spike. So we measure the gap between the spike. So this is example of homogeneous Poisson model. And we can also do a spike train autocorrelation to understand the distribution of times between any two spikes. So you detect patterns in spike trains like oscillation. So you can see the autocorrelation here and cross correlation here. So this is for cats, primary measure for cats. Cross correlation, there are meaning for this. A peak at zero means synchronous, and a peak at not zero means phase lock. I'm not going to go deeper into this step. It's enough that you understand that you can do autocorrelation and cross correlation to understand the distribution between any two spikes. Okay, next is population coding. So, so up to this point, we have focused on information carried by single neurons, but typically, informations are encoded by neuronal populations. Encoding by the most active neuron sounds reasonable if there is no noise, but it does not work in practice because of such fluctuations in neural activities. Basically, many neural systems use large number of neurons to input information. So that's why we do population coding. So when we study population coding, we must consider whether individual neurons act independently or whether correlation between different neurons carry additional information. Synchronous firing of two or more neurons is one mechanism for conveying information in a population correlation code. And the biggest advantage of population code is the ability to average out noises in individual neurons if they are independent. So these are these are two. This is some theory of perceptions in population coding on the temporal binding. Okay. Theoretically, this model assumes that neural synchrony with precision in the millisecond range is crucial for object representation, response selection, attention, and sensory motor integration. So, what it means is that neurons that respond to the same sensory object may fire in temporal synchrony. So, what does temporal synchrony means? We look at this example. You have four electrodes being put here. Okay, this is just an example. Yeah? So this is bistability. So all at the same position, but in left and right uh, um, of the background face. So if okay, in this case, the temporal binding model predicts that neurons should dynamically switch between assemblies, and hence that temporal correlation should equal for the two perceptual state. So we have four cortical neurons with receptive fields. So this is four, and then this is the result. So neurons 1 and 2 should synchronize if the respective contours are part of the one background face, and for 3 and 4 for the candle states. So when the image is segmented into two opposing states, the temporal correlation switch to synchrony between 1 3 and 2 4. Okay, so which means that how it responds, 1 3, the set of 1 3, these two will respond in synchrony with 2 4. Okay, so the same neurons that respond to the same sensory object may fire in temporal synchrony. So they fire in temporal synchrony because they are sensing the same object, which is the candlestick. Okay, there are three main steps for population analysis of neural coding. Okay, so this is the whole process. Okay, so this is the stimulus apple. The sensory here is visual sensory. So you see an apple, the process the action potential. So these are the spikes train produced, and then these are the process. They process this into they code into spike. These are spikes. The 111. These are actually spikes. So you can see just now, if you can see the spike sorting algorithm, it produce plots like this. So this is the plot that we process. So the first step is to record. Recordings are taken at different sites with implemented electrodes. So this you implant the electrodes on your brain. Okay, and then the stimulated activity of single neuron extracted from the continuous data using spike sorting algorithm. Okay, and then so this is they extract the information using spike sorting algorithm, and we produce all these spikes. And then information is inferred from the multiple spike prints with decoding algorithms. So these are the multiple spike prints produced with decoding algorithm or with information theory. So this decoding algorithm, algorithms that can predict that the stimulus was an apple. So this can be data mining approach or machine learning approach. Information theory is just based on probabilities or calculations, numerical calculation, which quantifies the knowledge about the stimulus gained by observing the population response. So the population analysis allows the study of the information carried by different features of the multiple spike trains. So it can be established whether the information of the apple is given by an increase in firing neuron in red. So this one, by a particular temporal firing pattern neuron in green, and by the stimulus simultaneous firing of subset of neurons in blue and green. The vertical dotted line, this is stimulus onset. Okay, so when this is time and this is where it sees the stimulus. Stimulus onset means when the stimulus appear in your eye and you sense your visual capture the picture. Okay, so this is important for you. This is very important. Single BS neuron, single neuron BS population coding in one neuron. For independent spike code, you use rate coding is enough. For example, using Poisson process. And correlation code, the information is also correlation of two spikes, not more than 10% of information in big codes. Okay, so you can have correlation of two spikes. At least two spikes will give you an information already. In population, based on individual neuron, and the correlation between individual neurons adds more information. So these individual neurons can work in either in synchrony or rhythmic oscillation. Okay. Any questions so far?
No.
Yeah, go ahead.
No question.
No question, okay? Alright, so I'm going to continue information theory. I'm not going to explain deep into this information theory so much, but I'm going to discuss on how it's being used for these cases lah. For spikes, to decode the spike trains just now. Okay, so information theory based on probability of observing a response. So we like to know the probability of observing an apple. Okay, so what is the probability based on the spike plot? The plot that we got earlier. What is the probability of observing an apple? So that is what we are trying to calculate using information theory, and the information is reported as in unit of bits, so temporal. Okay. Okay. So what is important is in information. This is how you could from this example above, the example of population coding just now, we only use apple, right? So let's say we have 4 stimulus: apple, pear, and banana. So how does their data being kept? I mean, the spike traits are being captured. Okay, so the green one is pear, the red one is apple. Okay, the yellow one is banana. So for the green one, after we have done all the process of spike sorting and all that, we get this representation, and we change it. Since it's a temporal coding, so we change it into binary bits. You have seen based on the spike counts. You have seen this before. So we bin the spike into the length of 3 bits. Okay, So if you think the line have to be similar, so that you can each bit position is referring to the same time gap or time uh, or rate, spike rate. Okay, all right. So for example, the green spike the green one is for pair. This is the pattern and it's being um, temporary, and the temporal coding gives you this representation 011 and the spike counts is 2. Apple also have spike counts of 2, but the pattern is different. It's 110. Okay, so okay, so the source of emission, first source of emission loss in the analysis of spike rate is given by specification of the response. So for example, when the original temporal position is reduced, okay, when the spikes are being in time or when the total spike counts are used. Okay. So first, when information loss can happen before this, when they are converting from the original spike plot into this representation, we may have lost some information, and when we transform this spike plot into bin, uh, when this spike are bin in time, then we may lose information here as well. Okay, and if we just based on spike counts, okay, we can see that if we only use spike counts, you will lose a lot of information because if this spike count gives 2 and 2, but uh, we will need the bin information to distinguish between these two signals. Okay, so if we just take spike count, then we have lost a lot of information already to process. Okay, so in the use of decoding algorithms gives another source of information because decoder needs out information about unlikely stimuli. Given the response distribution of a division responding to apple, pear, and banana, when the neuron fires a response R1 just above the average response to pear, a pear will be decoded. However, this particular neural response not only informs us that the pair is most likely stimulus, but it also informs us that the banana is very unlikely. Similarly, when the neuron fires a response R2 just below the average response of two pairs, a decoder will again predict that a pair was represented. But it will miss the information that it is very unlikely that an apple was represented. Okay. So we're talking about R1 and R2 here. Okay, R1 and R2 here. So so if R1 is just above the average of response to pair, then it will say that we are looking at the pair picture. However, it informs us that that is a pair, and it is if it's above average, then it's very unlikely that it is a banana. So when it sees at this point, okay, above average, it sees pear, and definitely it's not going to be banana. But when R2 is fired, okay, you again predict pear because it's just below average response pair. It's very close to pear. So we'll say that, yeah, this is a pair, but it doesn't say that it's not an apple. Okay, so there might be a confusion for us whether this pear or apple. Okay, so at this stage, pear and banana, okay, definitely pear, but not banana. Okay, this is definitely not banana. This is a pear. Okay. So more generally, information is lost when mistakes about the neural response probabilities are made. Shown the spike rasters of a simulated in single neuron to the three different stimuli. So this is the raster part. If the observer has to decode the response in time window 1, but does not know the precise post-stimulus time at which the considered response was emitted, it might set the decoder using the wrong response probabilities. For example, those corresponding to window 2 rather than window 1. Okay. The stimulus reconstruction will be then flawed, reflawed, and information will be lost. Okay. So we have window 1 and we have window 2. The one that you mentioned is not a mistake about the true responsibility are mentioned on the spike resters. You only have to get the response in time in one, but does not know the precise post-stimulus time at which considered response is you might set the coding. Okay. So if you put this on raster plot, okay, you could see that you don't know if you don't know where is the post stimulus and the stimulus onset, then you can see the information. There's a lot of information here, and you would not know. You have a different wrong probabilities. Okay, when you and you cannot reconstruct what is the stimulus. Okay, so based on the information loss. Okay, this is the next step for information loss. If you convert it into raster plot, then it's very it has huge information loss and further cause confusion. So you will cause wrong response probabilities. Okay, that is why if you look at brain injury patients, if they are or sometimes in the state of dizziness, you will not be able to tell things right or to see things right. Sometimes kan kalau when you pengsan ke apa, people will ask you how many how many fingers they want to do, and they show you your fingers and they ask you how many fingers how many days. So that's to make sure that you got the right your sensory is working right, so your response is also right. So in this state, if we decode this, this is an example of information loss. Okay, entropy. Okay, entropy. Entropy is a measure of response variability. I'm not sure why I have this slide here. Okay, so this is just for self-information. I think this is notation, just ignore the slide. Okay, let's look at mutual information. So entropy is a measure of response variability where we have mutual information in which we compare responses obtained using different stimulus on every trial with those measured in trials involving repeated presentation of the same stimulus. Okay. And the mutual information is the difference between the total response entropy and the average response entropy on trials that involve repetitive presentation of the stimulus. So how the entropy is being calculated, I'm not going to go through in depth because this is beyond you punya I mean it's enough that you know the high level, you know, you don't have to go to low level to calculate the entropy and so on. Okay, so but you need the entropy is just to compare response using different stimulus on every trial as compared to trials that use the same stimulus. For example, you have you have experiment, you have one control experiment. Okay, you let the monkey just watch at apple, and then you have the representation for apple only, and then you let the monkey watch the banana, and you have a representation for banana. And then what you do is you let the monkey look at number of number of pictures when you to banana, apple, pear, and so on. And then you decode, you take the representation when the banana appear, when the apple appear. So you take that, you compare to the representation of the one that you get earlier, the stimulus that you get when you show only apple, apple, apple. So that's the mutual information ID. Okay. So the repetitive is the when you show the same stimulus many times. So it's like you have a benchmark pattern for apple, and then you let the monkey see many pictures, and you see whether by giving many pictures, okay, the monkey will produce the pattern similar to benchmark pattern when it sees apple again. Okay, that is calculation. Okay, so basically that's mission information. And how all of these things being used in the computer science field. So we have data mining and machine learning methods to actually predict the so you remember this picture where we have okay, let me show you. Okay, so this picture, remember this? Okay, so we have covered information theory in bits. We use mission information and how we determine what it is, the probabilities of this being seeing an apple or seeing a banana using information theory. Just now what I show you. The decoding part is based on data mining or or machine learning approach. Okay, so now we look at decoding. Okay, so this is a general flow for uh, some research lah, but this is the almost the general flow. So you have the EEG data set, which is raw data set. You do segmentation and data preprocessing. Data preprocessing may include what we have learned, what we have saw earlier. Later I'll recap this. Random segment selection, generate star flow or random segments, quad tree. These are all computer science representation, and you get a pattern. Apply filter model for channel selection, select classifier, and evaluate the performance. So this is a general framework for overall. Okay, starting from the raw data, pre-process to get the plot and so on, and from the plot, we proceed to the algorithm, and then the algorithm either it says either if we do machine learning, and it's supervised, so it's either yes or no, so it can tell you whether it sees an apple or not. Rather than probability, will tell you yes, it sees an apple, no, it doesn't see an apple. Okay, this is step of pre-processing. This is what we have seen earlier. So we pre-process, and this plot, this plot is what we put into our algorithm. So this is data mining. Data mining is where we detect pattern. We have if this case happen, what are the pattern? Pattern 1, pattern 2, pattern 3. So we compare pattern. If this pattern appear in here, then there's this situation exists. So this is data mining. But to get this pattern, we need to do thorough experiments. Either by we can use the method, or we can use the information theory methods as well, or we can also based on running decoding methods. So just based and running the experiment time and time and try to find similar patterns. So data mining is about going through the whole. Let's say you have for each patient you have like 100 files, 100 files like this with signals for stimulus echo. So it will go through all the files to find the pattern. The computational algorithm will go through the file and find patterns, and they will suggest patterns like maybe like this or like this to signifies that we see echo and so on. Okay. So this is machine learning. Machine learning we need to know is a supervised approach. I think Prof. Putra would have explain to you how machine learning works. So machine learning is a label. You have to know what this pattern is corresponding to what, and then you divide the data set. Okay, this data set is being divided into training and testing set. Training set to produce a model to predict this pattern. So let's say you have a data set with all this pattern. So all this for this pattern. So you build a model to detect only this pattern, and then it will try and then we put in the testing data set. From there, it will use the model that we have. It will say whether yes it is yes or no. Okay, so that's machine learning. Okay, right. So that's the end of topic 3 and 4. I've shortened everything, but I've most of the supporting the supporting uh, the supporting materials are on the videos and so on to help you understand. Okay, right. So any question, open to question. So no question.
Prof,
Yes.
saya ada send gambar yang berkaitan dengan spike of data, spike of EEG punya reading kan. So ada send dekat dalam group WhatsApp. So sebenarnya saya nak tanya, is that spike itu adalah dia punya kekuatan brain, macam simplement dia adalah kekuatan brain yang ada pada kita punya klien tu ke?
Oh bukan, bukan kekuatan brain. Spike tu bila you proses, you akan nampak dia naik turun. Dia naik tinggi tu, kenapa dia naik tinggi? So maksudnya kenapa kan kalau signal tu dia kalau biasa sama je kan?
Betul.
Tiba-tiba ada spiking kan?
Ya, betul.
That one is action potential lah. Maksudnya ada berlaku something yang initiate the action potential. Okay. So but what initiate that? Patient tu tengah buat apa?
Patient tu dia bergerak.
Okay. So the action potential is telling the patient that whether it's from stimulus or is response. So kalau dia bergerak maksudnya it's response lah. Maksudnya the pattern that you get is the brain is telling the brain is telling the body to move. So that's the pattern for movement. What movement dia buat? For example dalam EEG ni orang kadang-kadang buat finger tapping movement.
Betul.
Ataupun eyelid movement of eyelid.
Yes. Okay.
So bila dia akan record lah. So dia akan bagi kat patient tu, okay, now you tap the finger. Then you can see the spike happening.
Oh.
Bila you see the spike happening, it's called stimulus onset lah. Maksudnya dia tap lah, dia finger tap kan. Bila finger tap tu berlakunya perkara tu.
Doktor, one more thing, bila kita buat neuro ni kan, dia ada banyak pattern, dia punya pattern EEG yang muncul. So adakah the pattern tu muncul disebabkan klien punya condition? Contohnya dia mengantuk, so dia punya gelombang dia akan lagi macam tinggi yang macam tu.
Yes. Kan ada I tak cover alpha beta, dia ada 5 bands kan, oscillation, yang tu oscillation.
Okay.
Oscillation, nanti you read lah on your own. Oscillation dia ada 5 bands, one of it ada drowsiness, arousal, semua tu lah kan. So kalau nanti kan EEG channel dia ada, kalau setengah software dia akan tunjuk berdasarkan wave tu juga, beta, gamma wave kan dia. So kalau betul appear dekat yang tu saya tak ingat lah mengantuk tu wave apa. Beta. Okay, so beta kalau dia appear kat beta maksudnya dia dalam keadaan mengantuk. Kalau dalam dia keadaan mengantuk tu, if the spike happen, what is it? Faham tak? Satu kalau ada studies yang buat untuk budak-budak belajar ni, the students belajar dalam kelas, daripada letak elektrod and they want to know dia orang punya fokus kan. So dari situ sensory dia adalah lecture tu lah, dia punya stimulus kan, dia ongoing stimulus, lecture tu ongoing. Tapi dia pun nak tengok student tu fokus atau tidak. So dia pun nak tengok lah dia punya wave tu jatuh mana.
Okay.
So itu jatuh, kalau dia mengantuk dia jatuh kat beta lah.
Okay, doktor, just macam oscillation yang doktor cakapkan tadi, macam alpha, beta, theta ni, kita ada normal range kan? Okay, bila melebihi sesuatu range tersebut, mungkin that's why lah menyebabkan kita macam drowsy, kita sakit kepala ke kan? So kalau kita as a normal client, bila kita buat EEG ni, and then the reading of the oscillation tu macam contoh usually alpha usually maybe dia tak normal daripada reading bacaan tu, is that kita punya brain sebenarnya tengah ada problem. Kalau kamu letak dia dalam representation graph ni, sebab kalau representation graph you akan nampak spiking lah kan. Okay.
Bila you nampak spiking, there's something is happening lah. So, tapi tak boleh tengok berdasarkan range je. Dia kena tengok, you punya ni. Macam action you punya gambar ni kan. Dia nampak mula-mula ni dia macam dia mendatar je, tiba-tiba dia naik. Tapi spike dia tak naik sangat kan.
Betul.
Tapi still ada spiking.
Okay.
So kat sini ni apa yang jadi? Apa yang you buat dengan klien ni? Sebab I cannot tell you. So
this client dia macam dia bergerak macam kuat jugalah dia nak ambil pen that time.
Okay, dia nak ambil pen. So yang tu lah you kena, yang spike tu yang you kena tengok.
Okay, baik.
So maksudnya dalam keadaan biasa dia punya bacaan sama lah, sebab tak ada stimulus kan, tak ada response. So dia tak perlu buat apa-apa. Habis tu bila dia bergerak aja, benda tu akan berubah.
Okay. Baik. Okay. Thank you, Prof.
Okay, next. Ada apa-apa soalan lagi? So nanti penting dekat bab dekat yang you need to understand the population coding, information loss. Itu penting. dan definition, type train, coding tu penting So this chapter penting this slide. Okay. Alright, kalau tak ada apa-apa dah, kalau tak ada apa-apa is I think may I know there is any textbook recommended? Okay. Alright, so June, I don't, because I have, if you look at the course planner, memang ada textbook. Tapi kan, the textbook is too thick. So I just extract information that is like, I just pick on information to the extent that I think you should know, and if you should not go deep, I don't go deeper lah for your case lah, because for all of this subject. Because if you go deeper, it's quite hard also to cover your different background. Okay. Yeah. Yeah. Boleh. Okay, so okay macam ni So paper tu kamu boleh basically conference paper memang dia akan ada sample result. So what you need to do is you modify the section headings lah sebab you buat literature review paper. So you can organize the paper according to your understanding. So first, of course, you need to organize it according to the subtopics that I gave. So for data format, data model, analysis and so on. So you at least you have the 4 subtopics. In the subtopics, you further organize your content. So when you read, if you see data formats, if there is a macam section that you can make out of it, then you do it. Okay, and then you put everything related into one section, and then you start to discuss. Okay, in that section, okay, let's say you say EEG. So data format EEG, format on EEG. So there's a lot of formats that have been suggested. You discuss one by one. You can make tables, and then you discuss, and then you critiques based on the advantage and disadvantage, and then you can say that at present the trend is using which format and why. That's what I mean by trends lah. I answer your question or is still not clear? Okay. Author your name lah. Okay. Okay. Kak saya nak buka e-learning je. Okay, share kat ni. Okay, e-learning kat sini kan saya ada bagi slide saya. Dulu saya cover this topic dalam kelas. Tapi since I've made it as your assignment. So I provide this slide for you guys to use as a guideline. So what kind of thing that you if you don't understand the assignment, you can refer to this slides. So we have data formats, data model, these data models, and then we have analysis method. So you can go through, and this is your supporting material for your assignment. Okay. Right. Any other question or any other matters? Boleh buat lah so far. Assignment tak susah sangat tu. Okay. So in that, okay, we'll try. Okay, good. InsyaAllah. Okay, in that case, we end our class today here. So enjoy the rest of the day and enjoy your weekend. See you again on 5th of December. We'll cover the last topic, which is topic 5, and then we'll have our test on 19th December. Okay, right. Okay, take care. Bye-bye. Assalamualaikum warahmatullahi wabarakatuh.
Doctor, thank you.
Thank you, Doctor.